Hey everybody, welcome to my studio in the basement of my house. This is where I'll be conducting all of my pre-recorded videos for computer applications class. This is also where I'll be doing all of the live video broadcasts for the scientific visualization class. Let me just give you a quick little tour here. We've got some diplomas and certificates and so forth, and some old computers hiding in the background. My skeleton, book collection, file cabinets, all the stuff that any professional illustrator is going to need. Um, if you've had a chance to read the syllabus, hopefully everybody has seen the revised syllabus. You know that I will be creating a handful of these additional pre-recorded videos. This is going to be particularly true when we get into, get deeper into Illustrator and start talking about some of the 3D effects like gradients and object blends and so forth. Let's see. Today's video is a continuation of something that I started very briefly before break. We had just a little bit of time to talk about the layout and organization and flow of illustrations, for didactic illustrations. So I'm going to continue that in a little bit more depth today. So I don't think you need to see me anymore, so I'm going to get out of this window and go directly into Illustrator. So one thing that we didn't talk about previously is obviously a lot of illustrations that are created in the medical and scientific field are just static views of structures. So obviously in medical illustration, a good example of this would be any kind of anatomic illustration. So you're really just trying to show what a structure looks like, identify the different parts of a structure, and so on, such as this skull view or this view of a heart. These are just static. They're not comparing anything. They're not showing a process happening over time. Other medical illustrations, of course, are going to compare and contrast different structures. And you've already done some of this with your cell illustrations. Here we see an example comparing different medical conditions involving the testis, like hydrocele or testicular torsion, which is actually a rotation of the testis within the scrotum, which twists up its nerve supply and blood supply, or varicocele, which is an enlargement of the spermatic veins within the spermatic cord. Now, one thing to notice about these comparison illustrations, and you've already applied some of this in your cell illustrations, is that things that are common to all of the different things that you're trying to depict in your illustration would be rendered using a similar style and similar colors. So in this case, the structures of the penis, the urethra, the erectile tissue, they're all rendered in with the same colors and the same style, since those things don't change from one panel to another in this comparison illustration. There are brighter colors, more saturated colors, that help draw attention to the things that are different between the different panels. Now, in this case, the colors are the same, so obviously, veins are always going to be blue, arteries are always going to be red. There's that structure called the epididymis, which collects all of the little spermatic ducts from the testis and leads into the vas deferens. All of these things are colored the same in the different panels because they are the same structure from one panel to another. But the fact that they are bright colors calls attention to them because many of them are important to illustrating the differences between these different medical conditions. Here's another example of a comparison illustration, and I particularly like this one because it allows you to compare many different things at the same time. The previous example, we were comparing just 3 different conditions of the testis. Your cell illustrations that you've created are comparing just 2 different cell types at a given time. But this was kind of a challenging assignment because I was asked to create a comparison between the shape and structure of normal red blood cells and the shape and structure of red blood cells in 4 different medical conditions. Now, I could have arranged these all in a simple column from top to bottom with the normal red blood cells at the top, and then the 4 different forms of anemia in little panels immediately below that. The problem that I had with that arrangement was that you couldn't compare, for example, pernicious anemia directly against the red blood cells because they would be separated by 2 or 3 panels. I chose this approach because it allowed you to compare the 4 different forms of anemia with the normal red blood cells, which are directly adjacent to it. Here's another unique sort of comparison illustration showing different forms of ectopic pregnancy. It shows the site of normal implantation of the embryo inside the uterus, but then it also shows ectopic locations or other locations where the embryo might try to implant. There is the tubal location where the embryo actually implants within the uterine tube or fallopian tube, and as you can see from the number there, that's 95% of all ectopic pregnancies are tubal. But sometimes the fertilized ovum can actually fall out of the fimbriated end of the uterine tube and implant directly on the ovary and begin to establish a blood supply to the surface of the ovary. Or the fertilized ovum might actually fall into the peritoneal cavity and implant either on the visceral peritoneum that covers the surface of the uterus or the parietal peritoneum, which extends down into the false part of the pelvis. So this is an interesting way of doing a comparison where it's not so much a comparison of different types of embryos, because the embryos in this case are all essentially identical. It's just comparing different locations within a single illustration. One thing we did start to talk about when I briefly introduced this topic right before break was sequential illustrations. So illustrations that are showing a process happening over time. And I think this is going to be particularly important for the process illustration that you're creating right now, the textbook illustration, which is designed to show some sort of biological process. And I think I did get to talk a little bit about some of the very, very basic principles about laying out these sorts of sequential illustrations. You want to think of them as being like a comic strip, where you have a series of panels. And it's very important how you organize the sequence of those panels. And in most Western languages, we are accustomed to reading from left to right and from top to bottom across the page. So for the majority of your viewers, they are automatically going to look towards the upper left corner of any illustration, assuming that that's the starting point. And their eyes will then gaze across the illustration from left to right or down the illustration from top to bottom, ultimately ending up down in the bottom right corner. That's just the way people are likely to read your illustration, as if it were words printed on a page. And so by following that flow from upper left to bottom right, the reader is much more likely to be able to follow your illustration. Now, in this particular example, it's not really divided into a series of separate panels, but the sequence of steps in the attachment of the HIV virus to the surface of a T cell is happening in a flow that goes not only from left to right, but to a small extent also from top to bottom. You know, the virus starts out up above the surface of the T cell and then is gradually brought down towards the membrane of the T cell, and then eventually actually fuses with the membrane of the T cell to insert the virus's genetic material into the host cell. So this illustration has both a left to right flow and a little bit of a subtle top to bottom flow. Here's another illustration showing a simple left to right sequential flow. This one resembles a comic strip a little bit more, where you have a series of 4 distinct panels that are flowing from left to right. This is showing the steps in angiogenesis, where VEGF, which is a vascular endothelial growth factor, stimulates a blood vessel to produce a branch, a new side branch. So there's the generation of this thing called a tip cell or a leading cell, which then initiates the process of formation of a new branch. And you see those steps here shown clearly from left to right. I think I also got to show you this example before break, showing a simple top to bottom flow where we have the development of the hematopoietic cells, the cells of the blood. So we start out with a stem cell that then differentiates into a series of different blasts, and these then proliferate further and differentiate further, and eventually give rise to both the red blood cells, the platelets, and all of the different kinds of white blood cells within circulation. And I think I made this point before break also, that you notice the colored background behind the cells. That's a colored background that actually has some meaning. I think I may have mentioned in one of your classes that I'm generally not a big fan of adding colored backgrounds to an illustration because most of the time I think they're gratuitous. They serve no purpose. But in this case, the yellow background represents the steps in this process that take place within the bone marrow, whereas the pink background represents the steps in the process that take place after the cells have migrated out of the bone marrow into the blood circulation. So in this case, background colors actually serve a purpose. Another example of top to bottom flow in an illustration, just showing some different things that trigger immune responses in a cell, and then some of the different responses to those different immune stimuli. And you can see that it's just a series of pathways flowing from top to bottom. Now, oftentimes, your steps in your sequential illustration will be arranged in a grid pattern so that you have both a left to right flow and a top to bottom flow. Now, this comic strip illustrates how readers typically read these kinds of multi-panel illustrations, where assuming that you have a uniform grid like this, where we have, in this case, we have 9 boxes that are all identical in size and shape, and they're all organized in a nice clear grid pattern, the eye is most likely to read it from left to right across the top, and then skip down to the second row and read that from left to right, skip down to the third row and read that left to right. Now, by the way, I'll point out to you that this particular image comes from this article, and I've posted the link for this article in your revised syllabus on my courses. And it's an article that's all about the flow of sequential illustrations. Now, in this case, they're talking all about comic strips, but comic strips are great resources or great metaphors for creating didactic illustrations, because comic strip is communicating information. Now, in the case of a comic strip, it's communicating a story, but the reader has to be able to easily follow the sequence of panels in the comic strip. Now, something simple like the example I just showed, where you have a simple 9-panel grid, it's very easy for the eye to follow the sequence of the story. But this article is interesting because it goes on and talks about lots and lots of other kinds of layouts that don't necessarily involve just a simple, straightforward grid with 4 panels or 8 panels or 9 panels or whatever the case may be. And it even talks about how different elements within the illustrations can help lead the viewer's eye from one panel to another, to help the reader follow the panels in the order which the illustrator intends for them to follow them. So this is something that Glenn actually talked about during the graduate orientation. I'm not sure if you remember that or not, but he was talking about things about how the way that a human face is facing, the reader is going to tend to look in the direction that face is looking. So if you draw a picture of a human face and the eyes are looking from left to right, the reader, when they look at that, they're going to follow the gaze of the person's face in that illustration to lead them to the next step in the process. So you can use all sorts of visual elements in your illustration to help the viewer look in the correct direction, or help direct their gaze from one panel of the illustration to the next, or one step in the sequence, one step in the process to the next. So let's go back into Illustrator. This is another image that was taken from that same article that I just showed you. And I found this very interesting, that this is actually showing some research that was conducted on how people actually read comics. And the article talks about the fact that if you have a simple uniform grid with just 4 identical panels or 8 identical panels or 9 panels or whatever the grid is, it's very easy for the reader to follow the flow of the panels in a grid like that. But once you start deviating from that uniform grid and have boxes of different sizes and shapes, like these different collections of rectangles you see here, all of a sudden it kind of screws up the way that people are likely to read them. And I was very surprised to see, for example, in that upper left-hand example, that in their research, people were much more likely to read from the upper left box to the middle left box instead of reading from left to right. They were more inclined to read down through the left-hand column and then jump over to the right-hand column. So your illustrations may not be this complex. They may not follow these kinds of unusual grid patterns, but just be aware that the way you arrange your images on a page is really going to dictate the way people look at your illustration and the way they follow it. Obviously, there are a lot of biological processes that don't just have a simple beginning and end. They don't just start from the left and proceed to the right or top to bottom. There are many biological processes that are cyclical in nature, things like the Krebs cycle or citric acid cycle in metabolism. This is a simple molecular cycle. It's the role of folic acid in the production and metabolism of methionine, one of the amino acids. And I think it's pretty straightforward, pretty obvious that when you have a cyclical illustration like this, that it's just going to proceed in a circle. And there is no clear beginning or end. Now, if you're working with an author, and the author is writing a block of text that's going to accompany the illustration, there's a very good chance that the author is going to have to start describing this cycle at some point. So in this case, the author might start talking about methionine first, and then talk about how methionine is converted to that other structure, the S-adenosylmethionine, or SAMe. So be aware of when the author is going to start talking about the cycle. I mean, even though this thing just continues around in the cycle, and theoretically you could start describing it at any given point, if the author begins their discussion of the cycle with a particular structure in the cycle, a particular step in the structure, typically that's the one you would want to put in the upper left and then show the process cycling around from there. Here's another example of a cyclical illustration that would be very common in a biology textbook, a pathology or physiology textbook, talking about the cell cycle and the cytokines and kinases and things that are involved in triggering the progression of the cell cycle to move from an inactive phase of mitosis into an actual mitotic phase to begin the process of cell division. And so that's obviously a cyclical thing too, because a cell undergoes mitosis, then it enters these other phases, the G1 and S and G2 phases, and then the daughter cells undergo mitosis again and again and again. So it's a process that just continues as a cycle. Now, in addition to simple linear sequences, top to bottom, left and right, or these kinds of cyclical processes. There are some biological processes where there are multiple pathways all leading to a similar outcome simultaneously. So I'd refer to this as sort of a simultaneous process, or a series of processes happening simultaneously. This is a bit of an extreme case because we see that there are many, many pathways all leading to the same ultimate outcome. In this case, this is showing how there are different processes both within the body, such as things like neutrophils producing oxygen radicals, or the process of just normal metabolism in the mitochondria producing free radicals. But then there are things from our environment like UV light, smoking, pollution, and so forth that produce free radicals in the atmosphere. And ultimately, all of these things lead to DNA damage in the cell. So we have multiple simultaneous processes all leading to a similar result. Now, sometimes, if you're lucky, you'll have a sequential illustration that you can superimpose over an anatomic or cellular structure. So you may have a process such as this illustration showing the ingestion and metabolism of vitamin B12, where your flow is superimposed over the anatomy of the digestive system. So obviously, your mouth is up near the top of your body, and your esophagus flows down into your stomach, which flows down into your intestines, which ultimately flows down to the rectum. You can superimpose this sequential illustration showing what happens to vitamin B12 and how it's ultimately absorbed in the intestine, and then superimpose that over the normal anatomy from top to bottom in the human figure. Now, obviously, this is not meant to be a very realistic illustration of the digestive system. It's highly schematic, but it gets the point across about how vitamin B12 is ingested and how it bonds with other factors to help transport it through the digestive system. And then ultimately you have this receptor that helps the vitamin be absorbed into the hepatic portal system of veins and then be delivered to the liver and then eventually distributed throughout the body. Here's another example of a process that is superimposed, in this case over a picture of a cell, showing the flow of the process from the outside of the cell to the inside of the cell. This is a type of chemotherapy that a lot of people refer to as the magic bullet, because you can create antibodies that are very, very specific to proteins that are expressed on the outside of tumor cells. And you can then deliver a powerful poison specifically to those tumor cells that express that particular protein on their surface. And the poisons will then kill only those cells that are targeted by this antibody. So in this case, there's a small sort of skeletal model of this drug, which has been attached to an antibody specific to this CD33 protein on the surface of certain leukemia cells. And this drug is ingested into lysosomes. The drug is then removed from the antibody in a natural process inside the lysosome. The drug then enters the nucleus where it causes double-stranded DNA breaks and preferentially kills only these leukemia cells. And finally, one last example showing a process that's superimposed over an anatomic structure. This is a very common illustration that a lot of people have done, showing the process of diapedesis and chemotaxis of neutrophils. So you have some kind of injury or an infection that leads to inflammation. This results in the release of inflammatory mediators into the bloodstream. and this causes the expression of molecules both on the surface of the neutrophils and also on the surface of the endothelial cells inside the vessel, things like selectins and integrins, which will then cause the neutrophils to attach to the inside of the blood vessel, to begin to flatten themselves out, and then ultimately to squeeze between the endothelial cells of the vessel and migrate out into the surrounding tissue. And then they'll follow a chemical gradient using a process called chemotaxis to migrate towards the site of inflammation. So, I mean, you could break this up into a series of separate panels like a comic strip, but there's really no reason to break this up into separate panels. You can show the cell simply flowing through the blood vessel and show it undergoing this sequence of steps to marginalize itself, to flatten itself out, and then ultimately to penetrate through the wall of the blood vessel. Okay, now that we've talked a little bit about illustrating comparisons, illustrations that compare one thing with another, and we've also talked about sequential illustrations, showing processes that happen over a period of time, we can combine that information to create illustrations that compare sequences. These are very common in the medical and scientific illustration field, where you want to highlight the similarities and the differences between two different biological processes. Here's an illustration I created for a journal article many years ago, comparing the process of meiosis in two different organisms, the giant surf clam and the human. Now that might seem like kind of an odd thing, but the giant surf clam is one of many species of organisms that are commonly used for research into developmental biology. This would be similar to things like the fruit fly that's such a common model for studying genetic information and genetic processes. It turns out that this giant surf clam and the human have somewhat different sequences or somewhat different processes of meiosis, and it's important for researchers to understand those differences because it can impact how the cells develop later on. And so if we try to use research about the embryology, the developmental biology of the clam, and then try to apply that to a human, we have to be aware of those differences in the early stage of development, or in this case, during the process of meiosis and the development of the gametes. So you can see here that some structures, as with any comparison, some structures are rendered using exactly the same color and style. So the nucleus in the light purple, the centrioles, the mitotic spindle, the chromosomes, and so forth are all rendered in similar colors. And really the emphasis in this illustration is just on showing the difference in sequence of when those things appear in the process. And one thing that's also important too here is the production of the polar bodies at the end of the process of mitosis, or excuse me, of meiosis. Here's another example of comparing sequences. These are three separate panels, although they look as though they are just sections of a single block of skin. And at the far left, you see cells in a quiescent state where there's been no injury, no infection, no antigen introduced, and so you have a whole collection of different cells that are part of the immune system, but they're all in a sort of an inactivated neutral state. And then the other two panels compare the processes of innate immunity and adaptive immunity. So you either have exposure to an antigen or you have some sort of injury, in both cases leading to inflammation, and then that initiates a whole cascade of effects with either release of cytokines or recruitment of different types of immune cells. So we're comparing these two different sequences of processes that happen in these two different components of the immune system. Okay, so these are other types of very common illustrations in the medical and scientific field. Finally, the last type of organization or flow, if you will, of illustrations that I wanted to talk about briefly are what I call level of organization illustrations. Um, these are fairly common in sciences where you're showing how a structure at one level of organization, at one scale, is related to other structures at a larger scale or a smaller scale. This is probably the classic example of these level of organization type illustrations that probably every medical illustrator is going to be asked to illustrate at some point in their career, showing the relationship of the actual contractile elements, the thick and thin filaments inside a muscle cell, how those are organized into clusters called myofibrils, how the myofibrils are then packaged into muscle cells, how the muscle cells or muscle fibers are then organized into fascicles surrounded by a membrane called perimysium, and then how those muscle fascicles are then bundled together into an actual muscle. Now, in this case, the flow, if you will, of the illustration proceeds the other way, where we start in the upper left with an overview of a cut section of one of the limbs, and then you see how the limb has several different muscles surrounding a central bone in the limb. So this could be the brachium, perhaps. And then you see that a single muscle has been kind of extruded out from the cut surface of the limb, and this has been cut away to reveal some of the different membranes surrounding the muscle, such as the deep fascia and the epimysium. And then you see how a single fascicle from that muscle is extruded out further to show how it's surrounded by perimysium. And then there's an individual muscle fiber or muscle cell that's extruded out of that. And then finally that's cut away to reveal the myofibrils and then the individual myofilaments, which are the actual contractile elements. And then you see there's also then enlargement of the sarcolemma, which is the membrane surrounding the muscle cell. And you see how that has these things called T-tubules and terminal cisterns and so forth, which are important for the movement of calcium ions, which are the things that actually stimulate the contraction of the muscle. Here's another example of exactly the same thing, just a little bit different version of it. By the way, I should point out that the previous example I showed, which was all in grayscale, that was just a rough sketch for a finished illustration. I don't actually have the finished version of that. This is for a completely different project, a different book, but showing pretty much the same kind of thing, where we have an overview of the upper limb, focusing in on a single muscle. You then see how the muscle is subdivided into individual muscle fibers, and then there's a series of enlargements to show how you go from the muscle fiber to the myofibril, and then you see how the myofibril is made up of these contractile elements of actin and myosin. So any of these level of organization figures are really just designed to show the relationships between structures at different scales, different orders of magnitude, or different magnifications. One last thing I want to talk about today is I just want to remind you a little bit about sizes of illustrations. And we did get to talk about this very briefly just before break. Remember, I talked about how in the publishing world, there's a system of units of measurement known as picas and points. And a pica is 1/6 of an inch, so there are 6 picas in an inch. And then a point is 1/12 of a pica, so there are 12 points in a pica. And you may already be somewhat familiar with points, because points are the units of measurement that are used to measure the size of text. When we talk about 10-point type, what you're talking about is the height of a typical lowercase letter measured in points. So as I said, there are 12 points in a pica, 6 picas in an inch, and therefore there are 72 points in an inch. And um, publishers will divide up the printed page into uh, columns, typically, that are a certain number of picas and a certain number of points in width. So if I go over and grab the rectangle tool in Illustrator, and I click anywhere with the rectangle tool, I can create a box of a certain size, and I could enter this in points, or I could enter it in picas. So let's say I wanted to create a box that was exactly 20 picas in width. I would enter this as 20p and then 0, meaning 20 picas plus 0 points. And then for the height, I'll just put in some other number here, let's say 40p0, and click OK. And you see how that creates a rectangle of a certain size, and that would be a typical one-column width illustration in a lot of textbooks, being exactly 20 picas in width. And then the height can vary, but usually the maximum size of a printed page is about 56 picas in height. So here you see an example of a one-column illustration where we've got a series of 4 panels. This is all about placement of an endotracheal tube, through the mouth, behind the tongue, and then ultimately into the epiglottis and into the larynx. And it's talking about different angles between different structures in the head that anesthesiologists need to be familiar with in order to properly insert the endotracheal tube. But the important point here is to see that there's a very faint light blue box, or sort of turquoise box, that I've drawn around these shapes, and that represents the width of my one column in this printed book. Now you'll notice that the illustrations don't completely fill the width of the column, but they're pretty close to filling the width of that 20 pica column. So this is a good example of what we'd call a one-column illustration. And then the height can vary, like I said, as long as the height doesn't exceed what we call the live area of the page, as long as the height is not greater than usually 56 picas, which is the maximum height of the printed area of the page. Here's a typical two-column illustration. Now, once again, we have 4 panels, very similar to the previous illustration, and I suppose I could have stacked these all on top of one another, but there were 2 problems with that. First of all, if you look at the shape of each small panel, each one is kind of in a portrait orientation. Each picture of the mouth has this sort of portrait orientation. So can you imagine trying to stack 4 of those on top of one another? We'd end up with something that was very, very tall, and probably wouldn't have enough room for it in the height of our live area. So in this case, it made a lot more sense to place these 4 panels side by side. I think it also makes it easier to compare the 4 different panels with one another because they're side by side. I think it would be much more difficult if they were spread out vertically from top to bottom. This was actually for that same book about anesthesia, and it's just showing 4 different ways that anesthesiologists classify the shape of the tongue and the oral pharynx, which then influences how they approach putting in that endotracheal tube. Notice again that I've drawn a light blue box, which is my bounding box around the images. I don't recall exactly how big that is, but if I just redraw a new box that's about the same size, I'm then going to delete it, and then I can just click with my rectangle tool. It's telling me that the width of the box that I just created, it automatically enters the size of whatever rectangle I just created. It's telling me that the rectangle I just created is about 476 points in width, which is actually about, it's close to 480, so it's close to 40 picas in width. Remember, there's 12 points in a pica. So that's telling me that this box is roughly 40 picas in width, which would be the typical size of a 2-column illustration. Now you might also remember right before break, I talked a little bit about how I normally set up the size of my illustrations. I don't actually create the artboard at this size. The artboard that I've created here is the standard 8.5 by 11 page. But then once I go into that standard 8.5 by 11 page, I'll then take my rectangle tool, click with it, and enter the size that I want. Whoops. Yep. I enter it in picas, but it's automatically converting it to points, but that's okay. And so I've created a box that is the correct size. And actually, if I compare it, the box that I've already turned into a guide, the light turquoise box, looks like it's just a little bit smaller than 40 picas in width. So they may have had a slightly different column size. This could be something like, 39p6 or something like that. But anyway, I've created a box here which is exactly 40 picas in width, which would be my typical 2-column illustration. And then I can go to the View menu, go down to Guides, and say Make Guides, or as you can see over here on the right, the keyboard shortcut for that is just Command 5 or Control 5 on Windows. And that turns it into that little turquoise box or guide. That is not going to print. That's not an actual object in my illustration, so that will not appear in print. It will not appear if I export this image as a JPEG or something like that. It's just there as a guide to help me make sure that my artwork fits within the required size, in this case, to fit within a 40 pica 2-column width. So that's the way I typically approach creating a new illustration. Is, like I say, I'll always just create it on a standard 8.5 by 11 page, and then go in with the rectangle tool, create a box that's the right size, and use the View menu, Command 5 to convert that into a guide, and then just make sure that my artwork fits within that guide. So that is it for today's video. I'm going to have another one, which I may post a little bit later in the week, about color calibrating your monitor. That was something I had hoped to do in class as an in-class demo, but I think I can probably figure out a way to do it here in my home studio. So thanks for watching, and I'll be talking to you somehow in class. It may just be email, or perhaps we can even set up some one-on-one video conferences at some point. So thanks very much.